So we did changes in predator exposure, but not in diet-induced phenotypic plasticity in scorpion venom. So when considering the motivations and universal applications for this experiment, there is an inherent arthropod race which promotes unique ornaments and weapons via reciprocal selective pressure. In the case of the Australian rainforest scorpion, the scorpion, like other venomous animals, employs venom in both offensive and defensive situations. Essentially, the scorpion promotes a plastic response by modifying its venom composition, which in turn conveys a higher fitness and allows for the scorpion to produce the most optimal and least amount of venom in variable environmental conditions. Concerning universal applications, researchers gain a further understanding in toxicity as well as further insight as to the physiological changes in response to the venom. The model organism, as described before, is the Australian rainforest scorpion. The research question is, how does induced phenotypic plasticity affect the venom composition of the Australian rainforest scorpion during predator-prey interactions? And finally, for the hypothesis and predictions, for the mouse, we expect there to be a high degree of relative proportions of the predator active toxins with half of the scorpions that encounter the vertebral predator, or the stuffed mouse. And with the cricket, we expect there to be a high degree of prey active toxins with the scorpions that encounter the live cricket as opposed to the dead cricket. And then for the mammalian cells, we expect there to be predator active toxins, which are more toxic in mammalian cells, with half of the scorpions that encounter the vertebral predator or the stuffed mouse.
So the methods for this experiment is we're going to catch different scorpions and put them in different experimental scenarios and extract, analyze, and compare their venom. So that's our scorpion as we're doing. And next slide. So these are the different treatments. So a live cricket will cause the scorpion to exhibit offensive pressure, and it could change the venom. And a stuffed mouse could cause it to exhibit defensive pressure and also could change the venom. So these are the 4 types of environments in which a scorpion can be in. So put the scorpions in these 4 different experimental treatments, and next slide. And so we put them in for 70 days, or 42 days in the treatments, and we collected venom 70 days. There's 3 collection times. So one was before the treatment, one was after the treatment, one was after another regeneration time. So they used the square wave stimulator to stimulate the action potentials on the glands, causing the venom to be extracted. Next slide. They analyzed the venom for 2 things, for composition and toxicity. So for the composition, they just put the venom in a size exclusion chromatography. It'll separate the proteins based on size, and then they can put them in different protein fractions. And then they shot it with 280 nm wavelength of light to see the absorbance. For toxicity, they put the venom on human cardiac cells and on crickets to see how it affected vertebrates versus invertebrates.
Okay, the results from the experiment showed a difference of the absorbances for the 14 different chemical fractions, or venom fractions. At the beginning of the experiment, there was minimal variance, except for within fraction 2. After the 49 days and the second collection of venom, the fractions started to shift out a little bit more. Toxin fractions containing the larger protein peptides, which included fractions 2, 3, 4, 5, 7, and 8, generally showed a higher activity on mammalian cells. And from the relative absorbances reading, indicated by the light blue and dark blue lines, those had the highest absorbances, especially in 6 and 7, which indicated those venom fractions from those experiment groups had the highest absorbances. And after, at the end, of collecting after the recover period, venom fraction 2 had the highest absorbance from the groups involving a mouse and live prey. But a lot of the groups involving the smaller protein fractions, which it was indicated in venom fraction 12, had a higher absorbency in the yellow and red graphs, which were the offensive situations. The other graphs in this experiment involved just proportional amounts of the crickets being paralyzed within the venom fractions from 8, 9, 10, 11, and 12, which are all the fractions involving smaller proteins. Those had the highest relative proportion of crickets which were paralyzed after roughly 60 seconds. And the venom fractions, including 4, 5, 6, 7, and 8, had the highest amount of cell index drop for the human cardiac cells. And these other scatter plots also show and indicated a higher amount of invertebrate toxicity for the fractions with the smaller proteins, which are known to be more effective against them, and same with the other.
Alright, so for our discussion overall, we have talked about the organismal venom chemistry that's been a major factor in terms of fight or flight responses of survival within its environment, and like what it does and how it does to affect the not only the scorpion but like its prey itself. So what we've learned is that it changes throughout the possible threats within the environment whether it feels threatened or not known to feel, which means is that should it choose to fight it or should it choose to not fight it based on how much venom it has at that moment. We discovered in our results that when the most effective one is that it's a dead mouse, but of course it's like mouse is like it's top of the food chain, so when it gets the mouse it's like highest above the graph of like most venom used, therefore to like really complete the kill in a timely fashion matter, which is the fastest there is. And of course the evidence of a change to venom composition is increased risk, but like it's like a trade-off where when a scorpion does a trade-off of selling its venom, it's like trading off its life. Like it's not that it's too much venom for this one specific animal. However, there's a scenario like what if there's too much if it's too many animals gang up within the scorpion to a point where he has no more venom, that it could possibly die and like have to like retreat to another area. So it has to like keep in check of that factor, whether or not it'll survive its attack or prey. Depending on its environment, whether the weather changes, the fall, spring, or like summer, mostly summertime, is how like its venom changes through its environment and how much it can use to survive through that. Not only its environment, but other organisms trying to fight it and trying to fight it or trying to find them. So it's like it's going to be a predator or prey, depending on where it's going to go with it. However, due to its plasticity, sometimes where the changing is going to take longer than expected through insufficient variation. So what if there's not much variety within its environment, therefore it's not going to stay in its comfort zone where the venom is only respond to that specific part of the environment, just not only around it, therefore it's going to be in a disadvantage. Same, that goes in with time lag, because it has to recharge and it has to hide somewhere, so it will be pretty useless at that time. So there have been a lot of cases where the prey they've been finding gangs up on them, therefore they're already like out of the game from there. And the absence of indistinguishable offense, where it lacks offense because it doesn't have the venom as the weapon, therefore it's like pretty useless without it. So that's our discussion of how much like it's pretty vulnerable in some cases. Yeah. Questions?